Good afternoon, class. My name is Dr. Ali, and I'm a microbiologist. I'm going to teach microbiology. Today, we are going to discuss the topic of mycology. So, today, this is our topic today. We are going to discuss mycology. Now, mycology, as you all know, is the study of fungi. But just a little bit of information before we get into the subject. What and why is this called mycology? Why is it not called, the as in the study of bacteria is called bacteriology, or the study of viruses are called virology? Why is the study of fungus not called, fungology? It is called mycology instead. It is because this mycos is a Latin word for mushroom, and this was the first fungus that was discovered, and that is why it is called mycology. That's just a bit of general information for you. And starting with the topic, we'll first discuss the general features of fungi. What are the features of fungi? Now, number one, fungi are eukaryotic. Okay, fungi are eukaryotic. Now, what does this mean? This basically means that the fungi contain all the regular cell organelles that we know of. That means they contain mitochondria, they contain the endoplasmic reticulum, they contain the Golgi apparatus, they contain the nuclear membrane. So all these cell structures that are basically missing in the prokaryotes, to which the bacteria belong, they are all present in the fungi. So hence fungi are eukaryotic, containing all the regular cell organelles. Second point is fungi are aerobic. So they may be either obligate aerobes or they may be facultative aerobes, but there is no anaerobic fungi. Fungi are in general aerobic. Number three is they contain chitin in the cell wall. Okay. This question has been asked multiple number of times, how the fungi are distinguished from other organisms. It is due to the presence of chitin in their cell wall. So that's the third feature that distinguishes fungi. Now number four, they contain ergosterol in their cytoplasmic membrane. Fungi contain ergosterol in their cytoplasmic membrane. So that is the fourth feature of fungi. Okay, chitin in the cell wall, ergosterol in the cytoplasmic membrane. Okay, so these are the general features of fungi, and this is how they are distinguished from other organisms. Now coming next to the classification of fungi. The classification of fungi. Fungi are basically classified in three ways. Okay, number one is the morphologic. Number two is the taxonomic. And number three is diseases. Okay, these are the three ways in which the fungi are classified. The number one is the morphologic, based on the shape of the fungi. Number two is the taxonomic, which is based on the nomenclature or the names of the fungi. And the number three is the diseases, according to the diseases that the fungi cause. So let's go through these classifications one by one. Number one is the morphologic classification. Morphologic classification has basically 4 divisions. Okay, morphologic, 4 divisions. Number one is the yeast. Number two is the yeast like. Okay. Number three are the molds. And number four are the dimorphic fungi. Okay. So those are the 3 morphologic divisions of the fungi. Number one are the yeast. And what are the yeast? Yeast are basically just unicellular fungi. Okay, single celled fungi. And example of this is the Cryptococcus. Example of yeast is the Cryptococcus. These are basically single celled or unicellular fungi. Now, yeast like fungi. Yeast like fungi are also yeast, but they contain budding cells which look like structures that we actually see in molds, which are the hyphae. Okay, hyphae and mycelium, these are the structures that are actually seen in molds. They are not present in the yeast-like fungi, but the budding that starts from these fungi, that looks like a hyphae, and hence this is also called as pseudo hyphae. It's not a real hypha, it's a pseudo hyphae. Example of yeast-like are the very famous, very well known Candida. Okay, example of the yeast-like fungi are the Candida. The next one are the molds. The molds, these are the actual multicellular typical classical fungi. Examples, that is, they are grown at a temperature of 25 to 27 degrees and they contain hyphae. And what are hyphae, by the way? Hyphae are filamentous structures which may be either septate or non-septate or septate filaments, and when there is a cluster of hyphae, this cluster of hyphae is called the mycelium. Okay, so the individual filaments are called hyphae and a cluster is called the mycelium. This is what forms a typical mold. A mold or a typical fungus, multicellular fungus, is made up of hyphae and the collection of hyphae is called the mycelium. This is the molds. Now, lastly, we come to the dimorphic. The dimorphic fungi, they, as the name itself suggests, dimorphic means they are having 2 morphologies at 2 different temperatures. At a lower temperature, that is again 25 to 27 degrees, they are molds, and at a higher temperature, which is at 37 degrees, they are yeast. Okay, So at 25 to 27 degrees, they are molds. At 37 degrees, they are yeast. So at a higher temperature, they are yeast. So if you want to remember this easily, you remember this cold mold. Okay. At a cold temperature, they are mold, and at a higher temperature, they are yeast. Again, this question has also been asked in the exam, where they have asked that which are the following are the features of fungi, and one of the features they said, or which is wrong about fungi, they asked, and they asked one of the features was, they said that the dimorphic fungi occur as yeast at a temperature of 25 to 27 degrees centigrade. So this is how they have asked, and the answer, that was the wrong one, because the actual thing is they occur at 25 to 27 degrees as molds and higher temperature as yeast. Okay, so these are the four classifications, and examples of dimorphic fungi are, sorry, we did not mention the examples of molds. Examples of molds are mucor, rhizopus, absidia, aspergillus. Okay, and examples of dimorphic fungi are the histoplasma, coccidioidomycosis, sporotrichosis, and so on and so forth. There are many examples of the dimorphic fungi. These are three of them: the histoplasma, coccidioidomycosis, and sporotrichosis. Okay, so this is as far as the morphological classification of fungi are concerned. Number one is the yeast. Example: Cryptococcus, unicellular fungi. Yeast-like, also unicellular fungi, but the budding appearing as pseudohyphae. Example is Candida. Third are the typical multicellular fungi, which are called the molds. Example of these are the mucor, rhizopus, absidia, aspergillus. And the last one is the dimorphic, which are occurring in two forms at two different temperatures: cold temperature, mold; higher temperature, yeast. Examples of these are the histoplasma, coccidioidomycosis, sporotrichosis, and so on and so forth. Okay, so that is the first classification that we have learned. Now coming to the second classification, which is the taxonomic classification. Taxonomic. Okay. Now, taxonomic classification is based on the sexual spores of the fungi. Okay. The naming of the fungi is based on the sexual spores. Now, let us start with the phylum. What is the phylum that the fungi belong to? Fungi belong to phylum Thallophyta. Phylum Thallophyta. This is the phylum that the fungi belong to. Now, this Thallophyta has two families, which are the fungi and the algae. These are the two families that the Thallophyta is divided into: fungi and algae. Now, since what is the difference between the fungi and the algae? Algae contain chlorophyll, whereas fungi no chlorophyll. Okay. So this is the main difference between the fungi and the algae. Here there is no chlorophyll in the fungi, algae contain chlorophyll. And since we are discussing fungi here, let us not focus mainly on the algae, let's focus mainly on the fungi. Okay. So fungi, we are talking about the taxonomic classification. And as we said, it is based on the sexual spores. So number one is the zygomycetes. Sexual spore is zygospore. Based on this zygospore, the name is zygomycetes. Second one is the ascomycetes. Here the sexual spore is the ascospore. Okay. By the way, what does asco means? It means it is a sac-like structure. The spore is contained in a sac-like structure. This is the ascospore, and the family, the class is called ascomycetes. The third one is the basidiomycetes, and the spore is the basidiospore. Okay, and here the spore is actually contained on a basidium. Okay, so these are the three main classes based on the sexual spore: zygomycetes based on zygospore, ascomycetes, ascospore, basidiomycetes on basidiospore, and there is one more last class which is called the deuteromycetes. And what is the sexual spore here? There is no sexual spore here. No sexual stage, no sexual spore. This is called the deuteromycetes, also known as fungi imperfecti. Okay, fungi imperfecti, the deuteromycetes. Again, this question also has come. They have asked the fungi without sexual stage, what are they called? And they didn't ask in the choices about deuteromycetes, they asked fungi imperfecti, and that was the answer. Fungi imperfecti or deuteromycetes. This is the fourth stage based on the taxonomy of the fungus and based on the sexual stage, and there is no sexual stage in the last class. Okay, so that is the second classification. The third classification is the classification based on the diseases that the fungi cause. The diseases that the fungi cause, number one is the, by the way, what are they called? The diseases caused by fungi are called mycosis. So one is the superficial mycosis. Second one is the subcutaneous mycosis, and the third one is the systemic mycosis. Okay, so this is the third classification, which is according to the diseases that the fungi cause. And here the three classes are the superficial mycosis, subcutaneous mycosis, and the systemic mycosis. Superficial mycosis, as the name suggests, it is mainly belonging to the superficial layer of the skin, which is the skin, hair, and nail. Okay, this is mainly the involving the skin, hair, and nail, superficial layer of the skin. And here the example of these are Tinea versicolor. Okay. Second is the very famous dermatophytes. Okay. And third is the Piedra. These are the examples of the superficial. Second one is the subcutaneous. In this, these are subcutaneous fungal infections. They mainly infect the subcutaneous tissues, and from there they cause a burrowing of the tissues underneath, and there is massive tissue destruction with sinus formation. Okay, so this is the characteristic of a subcutaneous fungal infection. There is infection of the subcutaneous tissues, followed by tissue destruction, followed by sinus formation. Examples of this subcutaneous are the mycetoma, number 1. Okay. Second one, chromomycosis. And third one is the rhinosporidiosis. Okay. So this is the examples of the subcutaneous fungal infections: the mycetoma, also known as the Madura foot, chromomycosis, and rhinosporidiosis. Which brings to our last class in the diseases, which is the systemic infections, and these are mainly fungi which invade the bloodstream and reach various organs of the body, and they also include the opportunistic fungi, which are mainly infecting the immunocompromised individuals. Examples of these systemic are the histoplasma, and candida, then penicilliosis, blastomycosis. So these are some of the examples of the systemic mycosis: histoplasma, candida, penicilliosis, and blastomycosis. Mainly they are invading the bloodstream, reaching different organs of the body, and characteristically found in immunocompromised individuals as opportunistic infections. Okay, so this is as far as the classification is concerned.